So let me now turn it over to her for her talk titled Harnessing the Power of Super Resolution Microscopy.
Thank you for the introduction, Steve. Welcome, everyone. As the title suggests, I wanted to kind of give you some practical insights of how to either improve super resolution microscopy techniques, especially single molecule localization techniques that you're already using, or if you're just starting out in the field, what are some of the things to look for? So I wanted to make it brief yet comprehensive. So with that, here's the brief outline of my talk. So I wanted to give a very brief introduction to super resolution microscopy, how it has evolved, and focus in particular on single molecule localization, which has been around since 2006, ever since I was postdoc in SAMP Lab. And it's evolved quite a bit. There's a lot of literature. It is continuously evolving. So I wanted to give you the idea behind the principles that it uses. And one of the central principle of single molecule localization is this blinking molecules, or how do you photo switch the molecules so that you get the best resolution possible. And that directly ties in with optimizing your samples so that you prepared them to be able to harness everything that single molecule localization is able to offer you. And I'll give you several biological examples, both drawn from my own personal experience of imaging with other folks in the field, and also published examples. So it gives you an idea of what can be done with this technique. So briefly, why did super resolution microscopy evolve? So for the longest time, confocal microscopy has been sort of the standard for doing biological imaging, and there are several good reasons for that. It's very easy to use. You could do multicolor, destacking, be able to do live cell imaging, but one of the main limitations has been the resolution, which is basically you're limited to about 200 nanometers in XY and about 500 to 600 nanometers in Z. So if you want to study biological processes with structures that are below the diffraction limit, then that is a serious limitation. So one of the first techniques to sort of address this limitation was structured illumination, where it gave you twofold improvement in resolution. And for some biological applications, that worked out really, really well. But we still want to push the diffraction limit. So the second technique to get commercialized was stimulated emission depletion. It kind of gives you about a 5-fold improvement in resolution. And that's also great for certain biological applications, but single molecule localization techniques such as POM, FPOM, GSDIM, there are several acronyms that are out there in the field, but predominantly we refer them to as single molecule localization. You're capable of getting resolutions of 20 nanometers in XY and then 50 nanometers in Z, and that's what the Vitara Bruker's Vitara super resolution microscopes are based on. That's the technique that they're based on. So just to kind of give you an introduction to what the Vitara 350 microscope is capable of doing, we've set it up so that it's live cell ready for when the field is evolving, and I'll talk a little bit about different live cell applications and the probes that have been developed to be able to harness the power of single molecule localization. So the Vitara 350 is the fastest 3D super resolution microscope on the market. So as I mentioned before, you're capable of getting 20 nanometers in XY and 50 nanometers in Z. And one of the nicest things is it's been engineered so that we've taught about the different modalities necessary for doing biological imaging in super resolution mode. So it was designed to be used by biologists, by scientists. The software is also very powerful to use, and there's a lot of cutting-edge features that are incorporated into the system, so you can easily image a whole range of biological samples. So with that, let me just go briefly into the principles behind single molecule localization. The whole idea is that if you have a structure which is below the diffraction limit, and if you're labeling them with biological molecules, fluorescent dyes basically, and if you're looking at it in a traditional sense, it's going to be blurred because of the diffraction limit. Now, in order to circumvent this, what you can do is engineer your sample and then image it in such a fashion that you only have a few molecules on at any single time. So which means you need a way to be able to turn these molecules on and off and also be able to control the density of the molecules. That's where the photophysics of the molecules come in. And so if you repeat this process over and over again for several thousand or 100,000 frames, then you're able to get a structure which is greatly enhanced in resolution compared to the original diffraction limited structure that you're seeing. So in a practical sense, how do we implement that? So here's an acquisition on the Vitara microscope. And as you can see, we're recording from 2 simultaneous focal planes, which is what we call the biplane, patented biplane technique. I'm not going to talk too much about that today, but this is what enables us to resolve things in all 3 dimensions. So you can see here the molecules are kind of coming on and off, which is what we call blinking. And then slowly the image is being built up. So if you had, of course, all these molecules on at the same time, then you're going to get a blurred image. Whereas if you can control it, and I'll talk about the different techniques to be able to control this blinking, you're now able to get a super resolved image. So as I mentioned earlier, there are different acronyms that are being used. To a certain extent, they're differentiated by the type of dye or protein that you're using, but in a global sense, you're essentially localizing single molecules. So here's a comparison of what you can expect. On the left is a conventional image taken on wide field and compared with the super resolution image that we obtained on the Vitara microscope. So these microtubules are a very standard sample to image because they're something that everybody can recognize what the structure should look like, and if you're not able to recover really good microtubule samples, images from a microtubule sample, you know that something's wrong with the system. So it's also a very good standard to be able to use. Okay, so as I mentioned before, one of the key things to be able to do for using single molecule localization is that you have to get the molecules to blink. So in a sense, we have to be able to control the photophysics of the molecule. So here's a very simple Zablonski diagram. So if you're looking at it as a very traditional fluorescence mode, you're essentially exciting the molecules from the ground state to a vibrational state, but that essentially relaxes really fast. That's the order of picoseconds, and then you can emit it back to the ground state. So that's the order of nanoseconds. So that happens very, very fast. So depending on the intensity of the lasers that you're using, proportionally you can also do something called intersystem crossing and then transfer molecules over to kind of like a unstable triplet state here. And the molecules that are in the triplet state are very, very susceptible to oxygen that's present in the system, and they can destabilize the molecule and essentially photobleach it. So if you wanted to be able to control this blinking state even further, so you can put reducing agents in the system and then create this anionic state, which is a much long-lived state. So this is basically, the triplet state could be on the order of microseconds, whereas this one, the long-lived triplet state that you can induce could be as much as like a few milliseconds or hundreds of milliseconds. So if you're able to kind of control this process using buffers and also the laser intensities that you're using, then you will be able to control the density and the intensity of your molecules and thereby optimize your imaging conditions. So this is just a much more detailed schematic of that one, where basically you can use a redox system in the triplet state, you use either a reducing or an oxidizing agent, and then kind of set up this reversible loop to be able to continuously cycle these molecules from ground state to a triplet state to ground state, and they go through a certain number of cycles. And so what we essentially need is a buffer, instead of imaging in PBS that we would normally image or some kind of buffer, we actually need a blinking buffer that consists of a reducing and oxidizing system, so which we call as ROX. So the most common ones that are used that I've come across, there's some kind of exotic ones that are very specialized to the type of application that you're using. The most common ones used are beta-mercaptoethanol and beta-mercaptoethylamine, which is MEA. These are both thiol molecules, so which can act as reductants. And then here's a schematic for like a glucose oxidase catalase system, so that you're actually controlling, you're taking out, depleting oxygen, and then releasing it in a controlled fashion so that you can affect these transitions back to the ground state. So there's been a lot of, I've kind of given you 3 or 4 publications here. There's a lot of publications talking about the photo-switching mechanism and how do molecules blink. But I would suggest that you go and look at the references that I've given here, because that gave me a really good idea of how to, what kind of dyes to use, how to control the blinking characteristics, and especially if I have multiple dyes, how am I going to make sure that all of them work optimally in the same buffer. And things have definitely gotten a lot simpler than they were 3 or 4 years ago. I think a lot of advances are definitely being made in the field. Okay, so now that we understand the photo switching mechanisms for blinking, so now we're ready to ask the questions that we need to ask if we want to get started in imaging in single molecule localization mode. So some of the questions that we want to ask first, these might sound very, very trivial, but these are the questions I usually ask when people want to start imaging. They have a protocol that they're using for confocal microscopy or wide field imaging, and they want to kind of transition into doing super resolution imaging. First question is, what do you want to image? And is it cells, tissues, bacteria, viruses? If you're looking at cells or bacteria or viruses, they're thinner. They're a lot easier to work with initially, but with the tissues, there's a lot of potential in terms of the data that you can obtain, as long as you're willing to put in a little bit of prep to be able to optimize that. And I'll talk about tissues and clearing in a few slides. And then what's your current sample preparation technique? Do you use, what kind of dyes do you use? Would you be able to change them into something that's going to be much more photon rich? And do you want to use fluorescent proteins, or are you open to using dyes? Antibody labeling versus doing transfections. There's a lot of options there in terms of what you can use. So that brings us to what kind of dyes do you currently use. Do you mount your sample? That's very, very crucial. What kind of dish are you using? Is that number 1.5 cover slip bottomed? These are some of the questions that you want to ask. And finally, what are your expectations? Do I want to look at co-localization? Do I want to go deep in and be able to look at the substructure of something in particular, a particular protein that I'm focusing on? So once you ask these questions ahead of time and then be able to optimize it a little bit, then I have seen just amazing data come out the first time I've actually imaged on a sample. Okay, so what are some of the preferred organic dyes? So two classes of dyes that you can essentially use. Organic dyes are fluorescent proteins. And organic dyes are tenfold more photon rich than fluorescent proteins. And of course, you also don't need to, if you're just starting out, you don't need to invest the time and effort into making a fluorescent construct. So in terms of the 4 different laser lines that the Vitara 350 offers, in the 488 range, my first preference would be Auto 488. For 5621, there's Psi3B, which is a modified form of Psi3, which works really well. With the 640 laser, Alexa 647 is the best blinking fluorophore that's out there, and you've probably seen this a lot in literature. And then we also offer 750 laser, and I've seen that Daylight 755 and Alexa 750 actually work really well in that wavelength range. So apart from that, there's a lot of other fluorophores that are available, and in some situations, depending on the biological system that you're using, you can use these other ones and try to optimize it. So you're not limited. The ones that I showed you earlier are the ones that I prefer because I worked with them a lot in a variety of biological systems, and they're very, very dependable. But there's a really good review from Xiaowei Wang's lab in Nature Methods that came out in 2011. They've done a really great job of characterizing all the different dyes that they've tested, and they have the quantum yields and how they work with different buffers. So if you want to get a much more comprehensive idea of the dyes that are available, I would certainly suggest that you go look at that particular publication. So apart from that, you also have genetically encoded photoswitchable or photoactivatable proteins. The first one that kind of started it all was PGFP, the original seminal paper that was used for POM and FPOM. And it's an okay protein. It's not very photon rich. So the best one that's available right now, if you wanted to try super resolution imaging, is Embryos 3.2. I've used that recently, and it works really great. It's bright. It's a monomeric protein. Originally, the protein was actually a tandem dimer. It was huge, and it was hard to actually transfect in and be stable. And apart from that, the other one to use is Dendra. And I have a couple of examples that I want to show you where we've used in my postdoc, I used a combination of this is Dendrite II hemagglutinin, which is a flu protein, and PAM cherry, photoactivatable M cherry actin. So we wanted to look at the diffusion of these clusters of hemagglutinin and how the actin cytoskeleton would be able to affect that. And so one of the limitations of using the fluorescent proteins is that doing multicolor is a bit of a challenge. And so you have some options. So in terms of doing multicolor, you have a lot of options with the organic dyes. But if you're willing to kind of optimize it for the particular system that you're using, you can do that with fluorescent proteins as well. So here's a publication from Sam's lab using 3 different fluorescent proteins. So these are very spectrally close together. Dendrite HA, which is shown here in blue, PM cherry actin, which is labeling the cytoskeleton, and then PM Kate, which is labeling the transferrin receptor. So, and I have a movie here, which you can take a look at how the transferrin receptors are kind of forming these tracks on the cell membrane. So that kind of gives you an idea that you can also use fluorescent proteins for doing the imaging. So I want to give you a few more examples. So since I started off with live cell imaging, a few more examples of live cell imaging done with fluorescent proteins and dyes. So this is from Dr. Jörg Beresdorf's lab at Yale. So they recently published with Embryos 3.2 clathrin. And so you can see this was actually imaged at a really high rate. So this was imaged at 600 frames per second, and 2 seconds worth of data was pulled. And of course, this is imaged in PBS. So if you looked at data all at once, you can see it's blurred. So if you pull the data into 2-second intervals, you can start seeing the little clathrin-coated pit and how it's moving as a function of time. So if you wanted to image even faster, look at faster dynamics, the route to go would be to use an organic dye because you are getting 10-fold higher number of photons with an organic dye. And here's an example, again, from the same paper. So looking at Alexa 647 labeled transferrin, this was imaged at 1600 frames per second, and 31 milliseconds acquisition time worth of data was pooled and then kind of rendered here into a movie. So in this case, the cells were imaged in high glucose DMEM, and in order to get the molecules to switch, they used beta-mercaptoethanol and glucose and oxidase and catalase, and this was imaged at room temperature. So depending on the temperature, you can image faster or slower. I just wanted to give you an example of transferrin clusters moving along the length of the cell here. So here, we only imaged like 3,000 frames, and then you can kind of see the clusters moving along. In this case, I also used switching buffer in order to be able to capture the trajectory of this particular Alexa 647 transferrin molecule. So here's an example from Dr. Suliana Manley's lab from EPFL. So they did some live cell imaging of direct label cellular DNA. So the dye that they used was pico green, and the imaging buffer that they used was, it had ascorbic acid, glucose oxidase catalase, and labeled its media. And so this was done at 30 milliseconds acquisition time, and this was imaged live. And there's also some publications from Dr. Xiaoyu Zhuang's lab where they use mitotracker, lysotracker type of dyes to be able to do live cell imaging. So yeah, I definitely encourage you to go look at some of the literature, and there's definitely a lot of potential for doing live cell imaging, and if not, you'll be one of the first ones to actually attempt that. And then finally, as far as live cell imaging goes, there are some dyes that are coming out on the market now where they can be imaged in PBS. So they're organic dyes that can actually be imaged in PBS. So here's an example of a Vera cell labeled with claprin and then tubulin and then imaged in PBS. And in terms of, if you're using these tiols and you're imaging live cells, how toxic is that? So one of the things that people have harnessed is to be able to use things like ascorbic acid or glutathione that's already naturally present in the cell to be able to act as a reductant. So there are some options there. So now I want to talk about the different biological applications that we've been able to look at using this particular technique. So in terms of infectious diseases, looking at labeling E. coli, looking at cardiology type of model systems, whether they're cardiomyocytes or cardiac tissue, reproduction, and that's a Z-stack that I'll show you. Looking at imaging thicker samples like Drosophila, and how would you be able to optimize it for that? Just looking at basic, cell biology functions such as imaging Golgi or the nucleus or mitochondria. Mitochondria tend to be a very popular model system to image, and one of the other popular science field to look at is actually neuroscience. So here's, let's start off with looking at E. coli. So here's an E. coli bacteria. This was labeled with sci-fi. For this one, it was a very simple labeling technique. Essentially, incubate the viruses with a sci-fi ester, and then essentially spin it down to take out the free dye. So in this case, we were able to obtain this 3D image with a single plane acquisition. So since the Vitara 350 microscope is capable of giving you 3D resolution with about a 1 micron depth of field of view, you're able to capture all this in just one Z-plane without having to Z-stack your image. So the nice thing about it is you can see like dye on the cover slip, and you can also see the E. coli bacteria standing up on the cover slip. And this has been color-coded for depth, which is the red is the bottom and then the blue moving away from the cover slip. And this was courtesy of Dr. Thomas Zell from the MD Anderson Cancer Center in Texas. Here's one of our recent publications from Phil Santanglo's group at Georgia Tech, where they were looking at RSV virus. And when we were first working with them, what we were pleasantly surprised with was they were already using the right dyes to be able to jump into doing super resolution imaging. So they were looking at RNA viruses labeled by using multiply labeled tetravalent RNA imaging probes they called MTRIPs. So this is a very clever way of labeling RNA in viruses. So they labeled the the antisense probe with Cyto-AB, which is in green here, and then the F protein of the virus with red. And what they saw, they also looked at the N protein as well, and what they saw were differences in the F protein and N protein distribution in the viruses. So you can see here that they're kind of edge to edge laid out, the RNA and the F protein, whereas in the N protein there is a little bit more intermingling, and they wouldn't have been able to see this level of detail if they didn't have the resolution that the Vitara microscope was able to offer them. And I'm very happy to say that was recently published in ACS Nano. So closer to home here at the University of Utah, one of the groups here was looking at imaging in cardiomyocytes. So these are canine cardiomyocytes, and they are looking at the sarcomeres. And what they wanted to see was, what is the difference between a control cell versus a heart failure model, both synchronous and asynchronous heart failure model. So this is a cell that that came from a canine which actually had heart failure. So what they saw was, they saw a decrease in the periodicity or the regularity of the alpha-actinin, and also they saw these little longitudinal depositions in between the actinin fibers as well. And this has also been recently published in the Journal of Cellular and Molecular Cardiology. So this is one of my favorite images. So this is a sample that Mark Lessard from the Jackson lab provided us. So these are spermatocytes extracted from 14-day-old testes of mice, and then labeled for Alexa647 CYP3 protein. And then they essentially, he sent me these suspended, and we were able to deposit them on a number 1.5 cover slip, use a little adapter, and then put buffer on it, and then take Z-stacks. So this one took about, people usually ask, how long did it take to take this image? So about 2 minutes or so, took about 80 Z-steps, and then the nice thing is you can see a lot of structure that would otherwise be blurred if you're just looking at it with regular fluorescence microscopy. You're able to see the spaces in between the chromatin fibers. You're able to get a feel for the 3D structures. So again, Mark, of course, spent a little bit of time optimizing the sample prep, and most of it essentially was what he would have used for confocal anyways. The one thing he was very careful about was, the extent of primary and secondary antibody dilution and how he fixed the samples. So those are some things to kind of keep in mind, and then also post-mixing your samples definitely helps. So now coming to more thicker samples. So this is an image courtesy of Dr. Sean Spies, who is at Oregon Health and Science University. So this is looking at a whole mount of softball larvae. The green is labeled with the frizzle receptor, and the red is Dilite 649, labeling the intermediate filaments. So this is about 6 to 8 micron thick, about a 6 micron thick stack. So this was about 0.4 micron C-step sizes that we took. We did sequential imaging, as you'll find for most of the organic dyes, so you do have to image them in a sequential fashion. And we were also able to image fairly deep. This is a thick stack that we took, but we were also able to image about 200 microns into this particular prep. So in that case, when Sean was looking at this, he said, okay, I had to really optimize and block really well so that I didn't have a lot of non-specific staining, and then just make sure that the primary and secondary concentrations were optimal. The other thing that he also had to do was optically clear the tissue. And that's when I got introduced to scale buffer. So it's a buffer. It's a buffer from a paper that came out in 2011 from Miyawaki's lab. And so for them, they were interested in looking at mouse embryos. So they wanted to be able to image really deep into mouse embryos for confocal imaging. And so you can kind of see the difference between leaving the embryos in PBS versus scale. There's 2 or 3 different types of scale. This was scale A2. The composition that I have is scale U2. So you can see that your structure, your embryo in this case, is optically very, very clear. So we essentially used scale U2, did the prep that you'd normally do for confocal imaging, and then you just store your sample, incubate your sample. In this case, for Drosophila, since it's a little bit thicker, probably a day or so would be enough. If it's something thinner, a few hours would be enough to clear whatever structure you're interested in looking at. The one thing is, it is a reversible process, so you kind of want to keep it in scale up until the point you're imaging. And we tried imaging in just regular buffer versus, doing imaging in scale buffer. And what we found was, as long as the samples were incubated in scale buffer, that was great. You didn't necessarily need to image in scale buffer along with the switching components. Recently, I talked to somebody who said he had used Clarity to do this, and if you have other clearing agents. So that's something worth trying as well. So the key thing is you want to make the refractive index kind of uniform, and then that way you can minimize the scattering. And so we've employed this with other kinds of tissue. So this is rat brain slices, 30-micron rat brain slices, looking at vesicular monoamine transporter. So employed similar labeling techniques that you'd use for labeling rat brain slices for confocal imaging, but we incubated these in scale buffer and then, again, deposited them on a cover slip and then put imaging buffer on them, and then we were able to image them. So here, Dr. Chris German from the University of Utah was interested in looking at the differences in structures, these dopamine transporter and SNAP25 structures, with and without drug treatment in an addiction model that he has been using for a while. And of course, there's also the most common application would be cell biology, looking at nuclear structures or mitochondria. So this is mitochondria, looking at Alexa 568 complex 4 and Alexa 647 TOM20 in red, which are inner and outer mitochondrial proteins. And this was an image courtesy of Dr. Cliff Guy at St. Jude's Research Hospital. And these two are from a HeLa cell where they were looking at a mitotic model. This is a 2-color where you can see the green is a microtubule, the red is a mid-body protein. And initially, these proteins, before mitosis starts, they form rings around the microtubules. And as mitosis proceeds, you can kind of see cone-shaped structures forming. So in terms of the mitochondrial imaging, what Dr. Kluskaj ended up doing, again, is use his usual concentrations of primary and secondary, and this was fixed in PFA, and then finally post-fixed in about, I think, 2% PFA, and then here we took about 7,000 frames for each color, like a buffer comprising of MEA and glucose oxidase and catalase, and then pH to about 8. So the final application that I want to talk about is imaging in neurons. That's something a lot of people want to do, and this is a really great publication from Xiaohui Zhuang's lab where they were looking at actin and different actin adapter proteins. So actin in this case was labeled with Alexa 647 phalloidin, and then either the beta spectrin or deucin was labeled with Cy3B, and they were able to get some beautiful 3D periodic images of actin and these adapter proteins forming these structures along the length of the neuron. So this was two-color imaging. Dr. Jason Shepard here at the University of Utah extended that to 3 colors, looking at a rat cortical neurons. So looking at Alexa48 MAP2, which is in green here, which is sort of like your contextual marker that you're looking at. And then in magenta, Alexa555. So this is one of the, this is not one of the dyes that I had tried before, but for their particular system, it actually worked really, really well. So looking at Alexa555 ARC protein, and then the cyan is GluR1. That sort of does not co-localize with the ARC protein. So in terms of, multicolor imaging, we routinely do 2 colors is pretty routine. Doing 3 colors is getting to the stage where you just have to make sure that, you label the structure that you want the most resolution out of. You label that with Alexa 647 or something in the 640 range, and then the next structure would be 561, and then if you want like a contextual marker, then you would label that with a 488. And the reason for that is, as you saw with the Zablonsky diagram that I showed earlier, the photo switching of far red proteins as you move towards the redder part of the spectrum is just a lot easier to manipulate those photophysics than with the green ones. So with the lower wavelength ones, you need a lot more power to be able to photo switch those, which is something we thought about on the Vitara 350 system. So that's why we have really high-powered lasers, about 1-watt lasers, to be able to do the photo switching. So initially when this field started, people were like, oh, I'm having a really hard time photo switching, let's say Auto 488 or Alexa 488, but my Alexa 647 is really easy to photo switch. So kind of keep that in mind when you're trying to figure out what dye to use for what structure. So the usual combinations, the 488, 561, the 2 common dyes that I usually use are 647 and then Psi3B. And we also recently started using the 750 laser. So now you can do up to 4 colors sequentially. And for the most part, we figured out that there's a couple of 2 or 3 different types of buffer recipes that actually work well with all the different fluorophores that are out there, and you should be able to optimize them and tweak them a little bit and be able to get really good images out of them. So in summary, what I want to say is, sample prep optimization is really very, very crucial to be able to get good resolution with single molecule localization. I can't stress that enough. It doesn't take a lot of time because I've worked with a lot of people who wanted to kind of get started in this field, just starting and asking the questions, okay, these dyes are good, will I be able to switch them? What would it take for me to optimize this? Choosing the right fixation reagents. What I found was if you're doing cytoskeletal fixation, that's having a little bit of glutaraldehyde, especially if you're doing tubulin fixation, works really well. Looking at it in cytoskeletal buffer, and also optimizing the primary and secondary antibody concentrations. Most of the time, you don't really have to change your primaries, but secondaries you will have to optimize such that your sample isn't too bright or too dim. Usually, I say, okay, let's start off with whatever you're using right now, and then if it's too dim and that you're not getting too many molecules, then we can up the secondary concentration. Or if it's really, really bright, you're having a hard time switching it, then we can reduce it. And then post-fixing, after you do your secondary labeling, helps keep all the dye molecules together and keeps the system much more stable. And also choosing the right imaging conditions is very crucial. Just making sure that the imaging buffer is compatible with the dyes. You're using the right cover slip, the right laser powers to match the switching rate, the right acquisition time, and how many number of frames that you want to take. This might all seem overwhelming initially if you haven't done this, but believe me, once you actually start doing the imaging, it's fairly straightforward, and it does become fairly second nature. So with that, I would like to thank you for your attention, and we'll be happy to take any questions.
All right, thank you very much, and great presentation. So we have a number of questions coming in. They're across a pretty wide range, and I guess maybe we'll try to group them and go through it sequentially. Seems like there's a number of people who are curious about some of the basic things, but then also chemistry and the details of the experiment. So I guess maybe following up on your conclusion there. Someone's asking, are there classes I can take to get started, or how does one sort of get going in this if they haven't done super resolution before?
Oh, okay. Just to get started.
Yep.
So if they've already done wide field or if they've done wide field or confocal imaging, yeah, I mean it's fairly straightforward. I usually just say, okay, what's the protocol that you're using, and basically just recommend the 2 dyes that I would normally use. And if it's a possibility to use those, then you just go from there and then do the imaging. It is that straightforward, or sometimes it's not, but most of the time it is that straightforward.
Okay. Well, this may be sort of, you covered it a little bit in that, but how do you choose between using fluorescent proteins and organic dyes for the SML, and are there other alternatives?
Oh, absolutely. That's a very good question. So most of the time, about 75% of the time, people want to use organic dyes just because they've been doing antibody labeling anyway. So it's easy for them to transition into that. And then most of the people want to do fixed cell imaging. So that option is usually what people go for. But then there's a group of people who want to be able to, do things that are much more physiologically relevant, and then in those cases they use fluorescent proteins. But since we're in science, people have figured out that hey, we need to get the best of both worlds. So more recently, in the past 2-3 years, there have been these like halo tags, snap and clip tags that people might have kind of read about. So you can actually do transfections and then use small organic dye molecules, which some of them are cell permeable. So you can actually do you can do transfection in a physiologically relevant sense, but also be able to use the organic dyes to get high photon counts and still be able to do live cell imaging. So yeah, I'm really excited. I mean, I see a lot of advances being made in terms of the availability of dyes and proteins that people can use.
Okay, yeah, so maybe you preempted this one as well, but is super resolution used with snap and clip tags, and maybe you can give a little background on what those are for the other members?
Okay, yeah. Oh, yeah. I've seen it getting a little bit more popular in the last probably year or so. So snap and clip tags are basically these little protein motifs that you can kind of, just like how you would transfect, let's say, clathrin or something, you would attach these like snap or clip motifs to your construct of interest, and then you have a little organic dye that would recognize the snap or clip or halo tag motif. So you just essentially incubate those. So one of the labs that I'm affiliated with does C. elegans imaging, and people who are in the C. elegans field know how hard it is to do antibody labeling on worms. So they've been able to figure out a way where they were able to transfect, if they're looking at synaptotagmin, which is a neuroprotein, they can kind of do like a halo tag synaptotagmin, and then they can come in with like an Alexa 647 halo tag and just incubate the worms with that, and then you would just automatically uptake it, and then you have it. I mean, it's actually there, it's labeled, and then you just image as usual.
Okay. So there's a number of questions on live cells. Maybe let's do one more before we get into that. So in traditional diffraction-limited techniques, the limited resolution is also a function of the wavelength. And I guess the question is, do the longer wavelength dyes have larger diffraction limits in super resolution?
That's actually a very good question. So in terms of, in a way, yes, but what really determines for single molecule localization is basically your signal-to-noise ratio. So the precision of localization is based on your background versus your signal. And in this case, actually, the Alexa 647 is very bright. So it doesn't necessarily mean that using Alexa 647 will give you, in this case, might actually give you a much better resolution in the terms of how we're defining localization precision. But overall, in terms of the point spread functions that you're obtaining on your system, they're absolutely, we are not breaking in classical physics. They tend to be a little bit wider than, let's suppose, your 488 or your 561. So, like especially if you're looking at that. But ultimately, your localization precision is going to be determined by the signal-to-noise ratio.
Okay. So now we have a number of questions on live cell. And so I guess to sort of kick it off, what do you see as the main limitation for broad adoption of super resolution imaging on live cells?
I think the current limitation is basically the dyes, because you have a limited amount of, if you're looking at the fluorescent protein area, you have a limited amount of dyes or proteins that are available. And if you want to look at slower dynamics, that's great because you have, a lot of dyes or proteins that are able to emit enough number of photons as long as you're slowly imaging them. But if you want to start looking at faster dynamics, each dye is only capable of emitting a certain number of photons. I mean, they're either going to emit it all at once, or if you're going to image really fast, they're going to emit a certain, subset of that total number of photons. And if you are background limited, then those photons might not be enough. So, engineering or, developing dyes which have a high photon yield or proteins that have a high photon yield is the limitation. Other than that, I mean, we've got the hardware, we've got the software, and we can definitely address dynamics up until a certain temporal point, after which the dyes kind of don't perform well enough to go faster.
Okay. And maybe a follow-up to that, what's the best way to reduce background counts then?
Okay. The best way is, of course, if you can image towards the redder areas, kind of go towards 647 or 750, you'll have less autofluorescence, and that's one good way. The other one, if you're especially looking at tissues, clearing with scale buffer, things like that, does definitely reduce the autofluorescence. Really good blocking, just choosing the right blocking, like if you're using BFA, goat serum, whatever it is, that helps. And what we found was if you have some kind of, if you're using chloraldehyde fix, or there's some types of background that can be quenched either using glycine or sodium borohydride. So I've employed all those techniques to be able to minimize the background that I'm getting from whatever biological structure that I'm imaging.
Okay. So we have about 5 minutes left. I think we probably have more questions than we're going to be able to answer. But again, if you type in your question, we'll follow up with you afterwards by email. If you want to ask a question, you just need to pop out the questions box, which should be in your control panel, and type it in, and we'll get it. So continuing with the live cell, in live cell imaging, does the glucose oxidase and catase need to be internalized in the cell?
It does, because you do want the molecules to be able to have access to it. But these are small enzyme molecules, so I have not had any issues with them getting into the cell personally. So ideally, yes. I mean, if you're looking at glucose oxidase catalase or some of the thiols, you do want the glucose oxidase catalase is just a much more kind of a broader way, because you're essentially, even if it's not completely getting into the cell, as long as it's able to extract the oxygen and you know, be able to recirculate it, that's fine. But if you're looking at some of the other thiols, they definitely do need to be able to diffuse into the cell. And I have not had any issues with any of the biological systems so far getting that to work.
Okay, and then maybe just as a follow-on to that, what determines the choice of the buffer that is used?
It predominantly depends on, you know, you're doing live cell versus fixed cell. You just have a lot more options that's fixed cell because you're not worried about killing something. So that's one. And two, it's predominantly determined by the dyes that you're using. And for the most part, what we figured out was having glucose oxidase catalase in there, pHing it from like 7.4 to 8. Those ranges work really well. And then having a little bit of MEA usually works. And then in some dye cases, adding the beta mercaptoethanol actually, like in case of Cy3D, it actually helps to have BME in there as opposed to not having it when you have Alexa 568. Whereas with the Alexa 647, it's very forgiving. So basically, you just choose the buffer based on the weakest dye that you have. The strongest dye usually happens to be Alexa 647. So in that case, you're like, okay, this is going to work with anything that I put in. So let's see with the other 1 or 2 dyes that I'm using what buffer works well, and then just go with that.
Okay. So this is back to your presentation. The question is, was the fibroblast example live cell? And then what the second example? So I guess that means the image or the slide after the fibroblast cell. Were those live cell examples or were those fixed?
Okay, let me just pull it up.
Yeah.
Oh, these ones, yes, yeah, those were a live cell.
Okay, and the one after?
These were live cell as well, yeah.
Okay. And then another question sort of on the details. You say 600 frames per second, but your images are on the scale of seconds.
Yeah, that's a very good question. So when it comes to doing, like, you know, photophysics and single molecule localization, a standard way of quoting, you know, how fast a microscope or, you know, your camera is capable of going is quoted in frames per second, because that just kind of gives you a background, like a benchmark to start from. So I mean, if you're looking at EMCCD cameras, you're limited, right? I mean, you could go maybe 70 frames per second, whereas if you're using SCMOS cameras, you can actually go 600 frames per second. Now, that 2-second temporal resolution is basically, just because you're able to image at 600 frames per second doesn't mean in 1 frame you're actually going to get any biologically relevant data. This is where the dyes come in. If you're able to, you know, get a lot of information in 1 frame, that's great. But just depending on, you know, the dynamics that you're looking at and the dyes that you're looking at, that might not be enough information. So your true temporal resolution in the biological sense is the 2 seconds, because that 2 seconds worth of data is actually giving you, you know, an idea of biologically what's going on. But if you are not able to image at 600 frames per second, the fast movement of the molecules would blur anything that you'd be able, you know, in order to be able to extract useful information. So you do want to be able to image as fast as possible and then figure out the right binning, so to speak. So in this case, we binned it at 2 seconds to be able to actually look at the, you know, the clathrin or transferrin or whatever we were looking at.
Okay, very good. So I think we're out of time. So I'd like to thank you again, Manasa, for the great presentation and discussion afterwards. And I'd like to thank the audience as well. If you have additional questions, again, type them in, and also please take the survey as you sign out from the webinar. That'll help us make these more valuable for you. So with that, I will wrap up this installment of the webinar series, and we hope to see you back next month. Thanks, everybody.
Thank you.